Hello everyone, I hope you're all safe. This is Dr. Nerd, and today we'll be talking about assimilation of nutrients. Now, there are three locations of digestion of macronutrients in your gut, and that's the lumen, microvilli, and cytoplasm. Of course, when we're talking about lumen, we're talking about your gut lumen, and microvilli is the microvilli of your enterocytes and cytoplasm of your enterocyte. Macronutrients are divided into three major groups, and that's proteins, carbohydrates, and lipids. Your primary site of digestion and hydrolyzation of the macronutrients are in the small intestine. Of course, why are we talking about hydrolyzation? And that's because proteins, lipids, and carbohydrates are just for example, carbohydrates are polysaccharides. So they're made up of more than one monomer of the same or sometimes different, most of the time different actually, monomer. Okay, and how do you break that bond? For example, if you have two glucose units that are bound together by glycosidic bond, you break that bond by hydrolyzation. Now, carbohydrates. Carbohydrates in your gut can only be absorbed as a monosaccharide, so as one molecule, one monomer, not polymonomers. Now, there are a lot of different types of carbohydrates. Today we'll only be focusing around those four: starch, sucrose, lactose, and cellulose. Starch is obviously a polysaccharide, and it's usually found in a storage form. And the two storage forms of starch is amylose and amylopectin. Amylose, the difference between amylose and amylopectin, I'm going to show you right now a picture of amylose. This is amylose. A small part of amylose. It's a lot longer than this. This is a glucose unit. So up from here, first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. And this is the link between two glucose units, the glycosidic bond. Now, the difference between amylose and amylopectin is amylose is the unbranched version of amylopectin, okay? Or you can say that amylopectin is obviously the branched version of amylose. And it only contains alpha-1,4 glycosidic bonds. While amylopectin, this one, it has alpha-1,4 glycosidic bond, and that's you can say that it's the backbone of the structure. And then you have alpha-1,6 between the sixth carbon of one glucose unit and the first carbon of another glucose unit, and it's called alpha-1,6 glycosidic or glucoside linkage. I'm sorry if the picture is unclear. I was just trying to, demonstrate how it actually looks like, just so that you have it as a visual representation. And starch is usually found in amylopectin form and not amylose. As you can see, amylopectin is 75%, while amylose is only 25%. You also have sucrose, which is a disaccharide, and lactose, which is also a disaccharide, and cellulose. Now, cellulose is only found in plants, as you all know. It's a dietary fiber. It has a lot of advantages in our body. It helps for, the fibers assist your digestive system, keeping food moving through the gut and pushing waste out of the body. Now, digestion of carbohydrate. It usually starts in luminal digestion. You have two types. You have the salivary alpha amylase and the pancreatic alpha amylase. The salivary alpha amylase, as you know, it comes out of our salivary glands in our mouth, and the pancreatic alpha amylase will come out from the pancreas. It's released or secreted by the pancreas. Now, alpha amylase is an endoglycosidase, which means it's going to hydrolyze the internal alpha-1,4 glycosidic bonds and not the alpha-1,6. which means I'm gonna just go back to so this we're inside, like we're in the middle of the amylose structure and not at the borders. So this is not the first carbon, and this is not the first monomer, and this is not the last monomer. We're in the middle. So alpha amylase will break down those bonds, the alpha 1,4 glycosidic bonds. Okay? And glucose is not a product because, as we said, if you want, we're breaking it down from the insides and not from the beginning or the ends. So you're gonna have, for example, it's only gonna cut two, like it's gonna cut this link and this link. You're gonna end up with two monomers, or this link and this link. You'll end up with three monomers, like a disaccharide or a trisaccharide. Now, this is digestion of amylose. I tried to draw it and make it easy for you guys to understand. This is one monomer, two monomers, three monomers, four monomers. It's a polysaccharide, so it goes a lot longer than this. And this is the alpha 1,4 linkages. And if you cut it, as we said, by alpha amylase, you're going to cut the alpha 1,4 linkages. You might get, for example, these two, which is a maltose unit, because as you know, maltose is a disaccharide. Or you can cut it here, which is also an alpha 1,4 linkage, and you'll end up with a maltotriose, which is 3. It's a trisaccharide. And like, if you guys realize, it's not actually a glucose unit, because glucose would be only one monomer, which is why, as I said here, glucose is not a product. Now, digestion of amylopectin. Now we're, as we've mentioned before, that alpha amylase is an endoglycosidase, so it's not going to break the alpha 1,6 glycosidic linkages, but only the internal alpha 1,4 glycosidic linkages. What happened? Okay, so if you draw it as a simple structure, you might only cut it from here. Example, you'll cut this, you'll have this as remaining, which is a maltose. Or you can cut it from here and you'll get a maltotriose, or two cuts from here, alpha 1,4 linkage and an alpha 1,4 linkage here, and you'll get the alpha limit dextrin, which is the main product of digestion of amylopectin. Only amylopectin can give out alpha limit dextrin because it has one different feature from amylose, which is the alpha 1,6 glycosidic bond, which I drew. I made it clear, I labeled it here. I hope it's clear. Now, as you can also tell, that there is no glucose. It's only multiple monomers of glucose, which is maltotriose, alpha limit dextrin, and maltose. Now, that was the luminal digestion. Now, the carbohydrate digestion in the brush border would use 4 different enzymes. So if you want to memorize it, you need to know that the brush border enzymes are 4 for carbohydrate digestion. There is lactase, maltase, sucrase, and isomaltase. Maltase also has another name, which is glucoamylase, and isomaltase also has another name, which is alpha dextrinase. Now you can tell that the alpha dextrinase, it's easier to know which one will it break down because of the name alpha dextrinase, which comes from alpha limit dextrin. And here is where you predominantly produce glucose, because in the brush border, this is where absorption of the carbohydrate monosaccharides takes place. Now let's start with lactose. Now lactose, obviously, which enzyme is going to work on lactose? Lactase. Lactose is made up of glucose and galactose. It's a disaccharide. Now lactase, I kind of drew the enzyme here just to make it easier for you guys to understand. The enzyme is going to have, there's two active sites, kind of, like you can think of it as two active sites, one for each monomer. So glucose, galactose, and then this is where the bond is, the alpha 1 4 glycosidic bond, and it's going to be broken down to glucose and galactose. Glucose is one of the products, which is why brush border digestion has predominantly glucose as their product, but of course there would be other carbohydrate monomers. Now maltose and maltotriose will be broken down by maltase, both of them. Yes, both of them, it's fine. Even though that here it's two, but it comes from the same enzyme. The same enzyme can break it down. Maltose is a disaccharide made up of 2 glucose, and maltotriose is a trisaccharide made up of 3 glucose subunits. And maltase would break down those alpha 1 4 linkages to produce 2 glucose in the if it was a maltose subunit, and 3 glucose molecules for a maltotriose. Now sucrose and alpha limit dextrin. Now, as I said here, there's a sucrase enzyme and an isomaltase enzyme. What I didn't say is that in the brush border, they are found attached to each other as a sucrase isomaltase complex. Now, they don't function together. If there's a sucrose molecule, sucrase is the one that's going to break it down. And if there's an alpha limit dextrin, the isomaltase subunit is going to break it down. Okay? Now, of course, as I just said, if there's a sucrose molecule, sucrase would be the enzyme that's used to break it down. Sucrose is a disaccharide made up of glucose and fructose. So if you break down the bond between glucose and fructose, you would get glucose as a product and fructose as a product. Sucrase is also known to break down maltose, maltotriose, and sucrose. So if you have this enzyme missing, the maltase enzyme, sucrase would also be able to break them down. And isomaltase would break down the alpha 1 6 glycosidic bond. That's the difference between maltase and isomaltase. Maltase, here, we said that it breaks down the alpha 1 4 glycosidic bond, while isomaltase, because it's an isomer of maltase, it won't break down the alpha 1 4 glycosidic, but the alpha 1 6 glycosidic. So when you have an alpha limit dextrin molecule, you're going to break down the alpha 1 6 subunits, and you're going to end up, for example, if it's a 2, if the alpha limit dextrin looks like this, it's made up of 2, like the backbone is like a linear bond between a glucose and the glucose, and then this glucose is attached to another glucose, and this glucose is attached to another glucose in the amylopectin subunit, and it's broken down to this structure. The isomaltase enzyme is going to break down the alpha 1 6 here and the alpha 1 6 here, but not the alpha 1 4. So you're going to end up with what? A maltose subunit. This maltose subunit can either be broken down by sucrase or by maltase. And then you'd have glucose and glucose as the product. I hope that's clear. What's important to know here that brush border enzymes predominantly produces glucose for carbohydrate digestion. Now, the intestinal absorption of glucose, galactose, and fructose. I tried to get you the same, it is the same picture as the one Dr. Peter put in his lecture for the assimilation of nutrients. This is an enterocyte, okay? This cell. This is the brush border side. This is the basolateral membrane side. And here, you can pretend this is the lumen, the gut lumen. So, gut lumen digestion is going to take place, and then brush border digestion is going to take place. And then how is the glucose units glucose, galactose, or fructose, because those are the main products of digestion of carbohydrates. They will enter the enterocyte by transporters. Now, there are different types of transporters and they work in different ways. First, you're going to have this, you've probably seen it a lot, especially during MSK, and foundation actually, sodium potassium ATPase pump. Now, from the word, from the name of the transporter, you can tell that it is a active transporter. Okay. So what do I mean by active transport? It means that it takes up ATP, as is shown here. So 3 sodium molecules are going to leave the cell from the cytoplasm to the this is where you'd find the blood vessels, so it will go up into the bloodstream, and then 2 potassium molecules will enter the enterocyte, and ATP would be used in the process. And this is what we call the driving force for the movement of glucose or galactose, which is going to be taken up from this transporter, the sodium glucose transporter 1. Why do we need this? Because this transporter will take out 3 sodium molecules out of the cell, okay? And this enzyme, this transporter or carrier, it's a symptoter, it's a co-transport. So it takes in sodium. For it to be able to take in sodium into the cell, we need a sodium gradient. So this part of the cell, the gut lumen, should have a lower, I forgot that PowerPoint has a pen. This would be so much easier. It has a less sodium gradient than inside the cell, which is sorry, oh my god. This side of the gut lumen would need to have a higher sodium concentration than inside the cell, which needs to be low for a gradient force of the sodium ion to be able to enter the cell through the carrier. Now, this is why we call the sodium glucose or galactose transporter 1 as a secondary active transport, because itself does not need ATP to work, but it needs this carrier, the sodium potassium ATPase pump, to use up ATP to take out sodium out of the cell, for this carrier to take in sodium into the cell, and as well with it glucose or galactose. So it's a secondary active transport. This is a primary active transport because itself uses ATP, and this is a secondary active transport because it relies on this carrier, which uses up ATP for its own carrier to work. Okay, and this way you'd have glucose and galactose into the cell. So now you have glucose, galactose, galactose into the cell, and sodium, which would be used up by this carrier out and then in with it as well, glucose and galactose, and this is how this carrier works. And now you have the GLUT5 transporter. I'm pretty sure you took them in Carbohydrates chapter with mole. Not sure if you took it or not yet. You should be by now, I think. Not really sure, but if you didn't, it's a type of transporter which is a facilitated diffusion. By that means there's low fructose here, there's high fructose here, and fructose, to be able to enter this cell, it would just need this carrier. By the help of this carrier, it can enter. You can't see this, can you? Okay, no problem. Oh no, you can